Hello students, welcome to EPG Pathshala. I am Dr. Imtiaz Hassan from Jamia Millia Islamia. Today we are going to discuss about the module Current Trends Comparative Genomics under the paper Bioinformatics. So in the Comparative Genomics we are going to discuss about the basic overview of the comparative genomics, why we do the comparative genomics analysis, then what are the assumption and limitation of the comparative genomics, then the genome analysis and annotation of the standard procedure, what are the standard procedure for the genome analysis and derivation of the function from the genome, then the general purposes of the database for the comparative genomics, why the database are created and how the information derived from the database in the form of comparative genomics, then organism specific database, then the genome analysis environment, then genome sequence alignment programs, and finally the genome comparison visualization tools. So let's describe the comparative genomics. The comparative genomics is the studies differences between the genome sequences, pinpointing changes over the time and comparing of the number or type of changes against background such as neutral expected changes, provide a better understanding of the forces that shaped the genome and the trait. A comparison of the gene numbers, gene location, and the biological function of a gene in the genome of the different organism. One objective being to identify group of genes that play unique biological role in a particular organism. So the comparative genomics actually analyzing and comparison of genomes from the different species, and its very well defined purpose is to gain a better understanding of how a species have evolved, to determine the function of genes and non-coding regions in a genome, then the function of human genes and other DNA regions often are revealed by studying their parallel in the non-humans to see that whether the similar gene is located in the other non-human species or any other comparative species. Then researchers have learned a great deal about the function of human genes by examining their counterparts in a simpler model organism such as the mouse and then compare with the other living organism. So the features that looks at when the comparing genomes, that first is the sequence similarity, how the particular sequence are similar or the differed, what level these sequences are differing, then gene location, where the genes are located, then the length and the number of the coding region within a gene, then the amount of non-coding DNAs in each gene can also be compared to see the comparative genomic component of the particular two genomes, and then we can found the highly conserved regions maintained in an organism. Hence, to do all the analysis very tricky and it's very hard. It needs a lot of work. That's why the computer program that can line up the multiple genome and look for the region of similarity among them are used. And for that, many of these sequence similarity tools such as BLAST are accessible to the public over the internet that can be used to get the information about the gene by comparing at their genomes, and then you can get many fruitful results or many useful results from the comparison of the particular genes or the genomes. We discuss about the mice and main sequence comparison. You can see the full complement of the human chromosome can be cut into 150 pieces and then reassembled into a reasonable approximation of the mouse genome. The color of the mouse chromosome at the number alongside indicates the human chromosome containing homologous segments. This pairwise similarity between the mouse and human genomes means that the insight into the mouse genetics are likely to illuminate human genetics as well. So it means that in the comparative genomics, the human and mice sequences are highly comparable with each other. We discuss about some basic terminology, that is the homology and analogy. So homology is the relationship of any two characters, such as the two proteins have the similar sequences that have descendant usually through the divergence for a common ancestor character. Homologues are thus component of or the characters such as genes, proteins with the similar sequences that can be attributed to a common ancestor of the two organisms during the evolution. Means the homologous sequence having the similar in the origin but may or may not be differed in the function, but by origin they are the similar or they share the common ancestor. However, the analogous are the non-homologous genes that have descended convergently from an unrelated ancestor, and they have similar function although they are unrelated in either sequence or structure or evolution, but functionally they have the same. So the homology and the analogy must be cleared to understand the comparative genomics. We further discuss another terminology that homologous sequence can either be the orthologues, paralogues or xenologues. So what is the orthologues? Means the orthologues are those homologues that have evolved from a common ancestor gene by speciation, and they usually have similar function. However, the paralogues are the homologues that are related or produced by duplication within a genome followed by subsequent divergence. They often have different gene functions. So the orthologues and paralogues, the basic difference is that the orthologues have the similar function, however the paralogue have the different function. On the other hand, the xenologues are homologues that are related by an interspecies or the horizontal transfer of the genetic materials for one of the homologue. The function of the xenologues are quite often similar. We further give the definition from the biology. For example, SNP, that's known as the single nucleotide polymorphism, that's the single mutation surrounded by two matching regions, and the region of the DNA where the two sequences have diverged by more than one SNP. Then the large inserts in which the regions are inserted into one of the genome sequences, like the sequence reversal or the lateral gene transfer. Repeats are the form of duplication that has occurred in either genome, and the tandem repeats regions that are of repeat of DNA in immediate succession but the different copy number of the in different genomes. A repeat can occur at the 2.5 times. So these are some basic definition drawn in biology to understand the comparative genomics. We discuss about the basic problem of the genomics. First, the vast number of species and the much large size of some genomes makes the entire sequencing of all genomes a non-optimal approach for understanding genome structure. The second, within a given species, most individuals are genetically distinct in a number of ways, and these two problems and the potential for other novel applications have rise to new approaches which taken together to constitute the field of comparative genomics. So these are the basic problem of the genomics which needs to the addition of the comparative genomics to constitute a new field of the comparative genomics. We can discuss about the unknown protein problem in various groups. If you see this map, that's the data from the seed, that the percentage of unknown proteins encoded by the diverse genomes, and you can see that although despite of the fact that the E. coli having the very small genome, but still they have around 25% genes are the function of 25% proteins are unknown. Similarly, you can see about the percentage ratio of the unknown proteins or the unknown genes, function of the unknown genes in the different organisms like the lactobacillus, staphylococcus, chlamydia, acidobacterium, solidbacter, then pyrococcus, then helicobacter. Then in the case of human and Arabidopsis thaliana, the maximum more than 50% functions are unpredicted so far. So that's the actually necessity for the comparative genomics to compare the genomic sequences in order to discover the function. Now we discuss why do comparative genomics. So the first answer is that many of the genes encoded in each genome from the genome projects had not known or predictable function. Means if you discovered the new genes, so may or may not its function is available. If their function is not available, then of course to know the exact function of that particular gene, we must do the comparative genomic analysis to find out the at least some clue about its function. Then analysis of protein sets from the completely sequenced genome, that how many proteins are present, what kind of proteins are present, at what extent these proteins are present. Then uniform evolutionary conservation of protein in a microbial genomes, and you know that the 70% of genes products from the sequence genomes having homologues in different distance genomes. That's why the comparative genomics helps to compare the two genomes in a well organized manner. Then the function of many of these genes can be predicted by comparing different genomes of the known function annotation and transferring function annotation of the protein for better studied organism to their orthologues in a lesser studied organism. That's why if the information is well annotated in the other organism, then the less annotated organism can be compared to derive the more information on the genomes. Then the cross species comparison to help the reveal the conserved coding region. So it can also help to do the cross species comparison. Then sometimes you don't have any prior knowledge about the sequence motif, that's why that can be determined through the comparative genomics, and it have the different type of algorithmic analysis to know the comparative comparison between the two sequences. We discuss about the power of comparative genomics. Suppose you have an unknown plant protein, so the BLAST P search give you the various prokaryotic hits, and none of them have clear function. That's the dead end. No, this is the beginning of the comparative genomics. Predicts function via guilt by association principle. Like the genes of related function are associated in various ways, enzymes in pathways, protein in a complex, and whatever the gene associate, they probably do it. So the comparative genomics actually, first you can find that what are the genes similar to these query sequences, and that's based on that can be done by the using simple BLAST tool. But only the BLAST is not enough, so you have to do some more analysis like the enzymes. If it is enzyme, what are the pathways analysis, you can do the motif search, you can do the domain search. If you have the structure like that, you can compare to predict the function of the unknown proteins. Like here you can see the association of evidence, that the genomic evidence that the gene A, B, C, D are the gene of the clustering. Through the gene fusion, these A, B, C, D are the shared common regulatory sites, and you can see here the phylogenetic divergence. And in the post-genomic evidence, that through the co-expression studies or the protein-protein interaction studies, or through the organelle proteomics, we can get the idea that whether these genes are associated or not. We discuss about some assumption and limitation, that homologous genes are relatively well preserved, while the non-coding regions tend to show varying degree of conservation. Conserved non-coding regions are believed to be important in regulating gene expression, maintaining a structural organization of the genome, and most likely other possible function. And the other limitation is that cross-species comparative genomics is influenced by the evolutionary distance of the compared species. So the basic or the general procedure for the sequence annotation is the first, that the basic procedure to determine the functional and structural annotation of uncharacterized protein. For that we use the sequence similarity search first, that's the BLAST or FASTA, to identify the functional region in the sequence. If greater the sensitivity is required, then the Smith-Waterman algorithm based program are preferred with the trade of greater analysis time. And then identify the functional motif and the structural domains by comparing the protein sequence against ProCyte, or the blocks or the SMART. These are the tools used to identify the motifs in a protein. Then we can perform conserved domain database analysis, that we found do the what kind of domains in the present. Then the PFAM, that it reveals the idea about from which family this belongs. So all these analysis can help to locate or to assign the function to the given gene. Then We can predict the structural features of a protein such as the signal peptide prediction, transmembrane segments, coil regions, and many other regions of the low sequence complexity. So these can help to the primary structure analysis of the protein, and based on the primary structure analysis, we can derive a lot of information. Then we can generate secondary and tertiary structure if possible, because the secondary structure gives the idea what are kind of the secondary structure elements, alpha helix, beta strand, and coils, and where these are located. And then the tertiary structure, for that we must need a template or the protein, homologous protein with the known structure. So the structure is another way to derive the function to the protein, because the structure is more conserved and the structure decides function of the protein. That's why these are the analysis can be utilized to provide to assign the function to the protein of the unknown function. Then annotation actually transfer the annotation is the transfer of function information from a well characterized organism to the lesser characterized organism. It can use phylogenetic pattern analysis. It uses the phylogenetic pattern search tools such as the cluster of orthologous genes to perform the systematic formal logical operation and gene sets. So these are the tools, or these are the methods actually used for the comparative genomics to annotate the function of unknown proteins. We discuss about the flow chart of bioinformatics flow chart for the comparative genomics. First, people need to do the sequencing by using any method, especially nowadays the high throughput genome sequencing techniques are available. So you should first sequence the gene sequences, then the analysis of the nucleic acid sequences, followed by analysis of the protein sequences. Then the molecular structure prediction can be done to predict the three-dimensional structure of the protein. Then the molecular interaction, what are the other protein interacts with this protein, can be done through the molecular interaction analysis. Then the pathways analysis, like the metabolic and regulatory network. Then the gene and protein expression data can also help that at what stage this kind of protein or this kind of gene is expressed more. Then it can be used for the drug screening, so the ab initio drug design or the drug compound screening database, and the genetic variability. So combining all these methods can give you the better idea about the comparative genomics and the function prediction and the genome similarity, and the basic concept of the comparative genomics lies under these tools. We discuss the two-dimensional gene annotation. These first dimension are the molecular function, like that enzyme activity or enzyme classification number, or in terms of functional context, for example the enzymes of a pathways. Two-dimensional are good, one-dimensional is bad, because even an EC number function may be wrong if pathway are not there, or the pathway context may be wrong if the certain enzyme are missing. So these are the correlation between the enzyme classification number and the pathways, and the gen bank etc. annotations are the one dimension for the molecular function. So only the sequence comparison is not enough. If you add the function prediction and the pathways analysis together in a 2D way, then the more precise function can be predicted of a gene. We discuss about the genome analysis, like the variation in genome size. For example, E. coli have 4.6 megabase pair, and the Mycobacterium pneumoniae having 0.81 megabase pair, and Bacillus subtilis having 4.2 megabase pairs. On the other hand, the GC content can be analyzed, like the Borrelia burgdorferi having 29% GC content, and Mycobacterium tuberculosis having 68% GC content. Then the codon uses, then the amino acid composition, like the rich in the glycine, alanine, proline, arginine, or in the GC rich. Then similarly the isoleucine, phenylalanine, tyrosine, methionine, aspartic acid are the AT rich sequences. Then we can discuss about the genome organization, like the large single circular chromosome or the linear chromosome plus extra chromosomal elements. So these are the different genome analysis can be done in the comparative genomics. Discuss about the CG comparison between the genomes. like the strains of the same species may have the different types of the genes, or may have the different gene, of course, and many of the genes are the similar. Then how these are the closely related species, the distantly related species, like the list of Orthologues, evolution of individual genes, evolution of organism. We discuss about the comparative genomics helps to ask some interesting question like identification of similarities or differences between two genomes that may allow to understand how two genomes are evolved. Means you can discover the evolutionary information between the two organisms or two genomes, how they are evolutionary related or how these are evolutionary differed. Then why certain bacteria cause disease while the other do not cause disease. This kind of information can be derived from the genomic analysis through the comparative genomics. Then the identification and prioritization of the drug target, of course, because the more dangerous pathogens can be targeted first, and what are the pathogenic entity or what are the virulent entity hidden in a pathogenic organism, that can be identified only through the comparative genomics. We discuss what are the units to be compared in a genome. For example, in a gene or genome, we must compare for the two organism or more organism, that what are the number, means the gene number, or then the gene content like the GC content or the sequence content, then the location, means the position in a gene in the chromosome, then the gene order, then gene cluster. Genes are the part of known metabolic path are found to exist in a group or the linked gene. Then collinearity of the genes ordered is preferred as the synteny. Then a conserved group of genes in the same order in two genomes as syntenic group or as syntenic clusters. Then translocation, movement of genomic part of one position to the another position. You can see here the E. coli gene, the tryptophan, Operon in the E. coli, in the Haemophilus influenzae, in the Helicobacter pylori, in the Bacillus subtilis, Archaeoglobus fungitis, Methanococcus genesi, and the Methanobacterium thermototophicum. So the number of genes and the arrow direction of the transcription description of Operon. So you can see here the different types of gene are located at the different places in a different organism. Like you can see here the Trip A and Trip B are genetically linked separate genes in the Methanobacterium. So like that, you can see that the comparative genomics helps to identify the different linked gene in a different organism, and that can be compared further. We discuss about the comparative genomics tools, especially those bioinformatics tools which are help in the comparative genomic analysis, such as BLAST2, then MUMMER, then PipeMaker, then AVID, then comparison analysis of both nucleic acid and protein level. So we discuss and believe that you should know that the BLAST is used for the sequence comparison and the similar sequence identification. We discuss about the MUMMER. Is actually genome alignment tool which actually align the two genomes together, and is developed by Steven Salzberg group at TIGR, and this is published in the Nucleic Acid Research in 1999, and its updated version is published in Nucleic Acid Research in 2002. It's freely available at the TIGR website, and that can be used for the genome alignment of the two organisms or more than one organism to can easily compare the genome information in the form of comparative genomics. We discuss about the basic features of MUMMER. It is the algorithm that assumes that the sequences are closely related. The first assumption that the sequences are closely related, so the closely related sequences can be easily analyzed and quickly compares millions of the bases. That's the good part of these tools, and its output is in the form of base-to-base alignments and highlights the exact matches and differences in the genome. Furthermore, it locates single nucleotide morphism between the two genomes, then the large inserts, then the significant repeats, tandem repeats, and reversals. So By analyzing or locating all these things, this tool is very useful for giving the information about the comparison of the two genomes together in a very well organized platform, and the output are well organized format. We discuss some automated genome annotation tools such as GeneQuiz, that it's having the limited number of search per day. Then Magpie, it's the outside user cannot submit on sequences, but they can utilize the sequences. Then Pedant, that's the commercial version, allows for the full capacity. Then another tool is the Seals, which is a semi-automated tools. These are the automated tools which can be utilized for the genome annotation in the comparative genomics. We discuss the general databases that use for the comparative genomics, like the Locus Link, that's the ref sequence database of the NCBI. Then Pedant, that's the protein extraction description analysis tool. And the MIPS, then the COGS, that's the cluster of orthologous groups of the protein. Then KEGG, this the Kyoto Encyclopedia of Gene and Genomics, that gives the information about the pathways database. Then MBGD, it's the microbial genome database, having the information about the different microbial genomes. Then another is the GOLD, it's the genome online database, having the information about the different genomes. And the TOGA, it's having information about the TIGR or the MUMMR. Now we discuss the problems with the existing sequence alignment algorithm for the genomic analysis. Most of the algorithms were developed for comparing the single protein sequences or the DNA sequences containing a single gene, and the most algorithms were based on assigning a score to all the possible alignments, usually by the sum of the similarity or the identity value for each aligned residue minus a penalty for the introduction of the gap, and then finding the optimal and the near optimal alignment based on the chosen scoring scheme. So unfortunately, most of these programs cannot accurately handle long alignment. For the short alignments is okay, but for the longer alignment, the gap penalty and the scoring function compilation, all these things sometimes creates a problem. Then the linear space type of the Smith-Waterman variants are too computationally intensive, requiring specialized hardware like the memory limitation, or very time consuming, and hence the higher speed versus the sensitivity problem is, if you want to do the higher speed, then sensitivity decreases, like that. So these are the basic problem associated with the algorithm of the sequence alignments. We provide the information about the genome size or the comparative alignment tools. Like these are the different tools like ASIRIC, accelerated search or similarity regions in a chromosome, and their URL are given here. Then BLAT, then D-align, that's the diagonal alignment. Then DBA, that's the DNA block aligner, that's present on the Sanger website. Then GLASS, it's the global alignment system. Then LSHALL, pairs means locality sensitive hashing in all pairs. Then MegaBlast, then MUMMR, it's the maximum unique matches. Then PipMaker, it's the percent identity plot maker. Then SHAHA, it's the sequence search and alignment by hashing algorithm. And WABA is the wobble aware bulk aligner. So these are the different tools used for the comparative alignment and for the comparative genomics comparison. We discuss about the genome comparison and visualization tool like the ACT, it's the Artemis comparison tools, and it's the displays parsed blast alignment based on the Artemis or an annotation tool and present on the Sanger website. Then Alfresco, it's display the DBA alignment. PipMaker, that displays blast Z assignment. Interic, that displays blast Z alignment. Then Intronator, that displays WABA alignment. And the Vista, it's the visualization tool of the alignment. And finally the SynPlot, that display D-align and the glass alignment. So the display or the visualization is much important because it helps you to analyze the genomic information in a very well organized way or in the comparative way. We discuss the post sequencing challenges, like the genome sequence is just a beginning to appreciate the biocomplexity, and the sequence-based function assignment approach fails as the sequence similarity drops. Hence, the structure-based function prediction approaches are limited by the availability of a structure, association of the structure motif, and association of the function descriptor as a result of any genome. So genes of known function are the 40% and gene with the unknown function are 60%. So it's the challenge to minimize the number of unknown functions by using the different algorithm or different available tools of the comparative genomics. Now you have learned about the comparative genomics, so we can summarize again that the comparative genomics provide a powerful way in which we can analyze the sequence data, and genes are complex structures which are difficult to predict with the required level of accuracy and confidence. And the comparative genomics involves the use of computer program that can line up multiple genomes and look into the region of similarity among them. It will help to understand the genetic basis of the diversity in organism, both speciation and variation, events that are important aspect of the evolutionary biology. Researchers have learned a great deal about the function of human genes by examining their counterparts in the simpler organisms such as mouse and the other organism. So the basic concept of comparative genomics, because the all modern genomes have arise from the common ancestral genome, the relationship between the genomes can be studied with this fact in the mind. This commonality means that the information gained in one organism can have application in other even distantly related organism. So the comparative genomics enables the application of information gained from the facile model system to agricultural and medical problems. The nature and significance of differences between genomes also provide a powerful tool for determining the relationship between genotype and phenotype through comparative genomics and morphological and physiological studies. So the features looked when the comparative genomics are the sequence similarity, gene location, length and the number of coding regions, and the amount of non-coding DNA in each genomes, and the highly conserved region maintained in the organism. And the purpose of the comparative genomics is to gain a better understanding of how the species are evolved and to determine the function of genes and non-coding regions of the genomes. That's all about the comparative genomics. Thank you.